This video will cover Lecture 1, which is review of topics that you might be familiar with. Now this course, we are not a cell biology course, but it is a good idea if you have some familiarity with things, say the difference between prokaryotic and eukaryotic cells, also the basic structure and function of organelles, like what is a mitochondrion, what is a nucleus, those sorts of things. They will come up during this course at some point. But we are, of course, a class that's more concerned with biomolecules, so sort of the smaller scale of things. And it's often said in biochemistry that there are these 4 canonical molecules. And the reason we sort of say this is because these generally have this sort of monomer-polymer relationship, more or less. So that means that there's something small, right, a monomer, that can be linked together to make something much larger, the polymer. And this is certainly true with nucleotides, the monomer, which can be linked together covalently to make what we would consider nucleic acids. And examples would be things like DNA, RNA as well. Amino acids, again a monomer, can be linked together to make the polymer known as polypeptides, which would be the example we're going to spend most of our time looking at would be something like protein or enzymes, right? Monosaccharides, meaning single sugar, can be linked together to make polysaccharides. And you might sometimes call polysaccharides complex carbohydrates. That's fine, but a couple examples might be things like starch and glycogen. Glycogen. Now those 3 examples, it's a covalent attachment, right? And in all 3 of those examples, those monomer units are covalently attached. But this last one isn't really covalent attachment. That's why we call it analogous, meaning that there's a small thing that kind of interacts to become a big thing, but those little things are not attached through a covalent bond. And that's when lipids, certain specific types of lipids, interact together to make membranes. And just to be clear, this is non-covalent. And so we often call these the 4 canonical molecules in biochemistry, and it's pretty common. And of course, like this, just sort of take a tour of these and look at their structures and functions and put everything together and make metabolism. And that's certainly what we'll do in this course. I do like to bring up that, there are molecules that don't fit into these categories neatly, or are combinations of those. And so one example I like to talk about, for example, the secondary metabolites and a primary metabolite, just so we know what the term means. Primary metabolites are things like the process of glucose being broken down through glycolysis, right? It's an energy need that the cell or the organism needs immediately to stay alive. Secondary metabolites are things that enhance their ability to stay alive longer term, but they might not have an immediate necessity. That's kind of a loose definition, but sometimes how I think about it. So another example of those secondary metabolites would be something like the alkaloids. Alkaloids, things like, say, caffeine, nicotine as well, right? These are compounds produced by plants that they don't need immediately, but they enhance the organism's ability to stay alive because of pests, of leaf eaters and other types of organisms that might destroy them. So for us, we can enjoy coffee or a form of nicotine if you so choose. We can have those and they give us like a mild stimulant effect. But imagine if you're a leaf-eating larva or insect and you eat, your body weight in nicotine leaves a day, right? That's going to have a pretty huge impact on your nervous system. In fact, it's quite toxic to those organisms. So it's a defense mechanism, right? Another defense mechanism would be, say, antibiotics. And so those might be things like, maybe you're familiar with erythromycin, right? Ampicillin, right? Penicillin, lots of those types of things. These are compounds produced by some organisms, whether it's a fungus or another bacterium, and they eliminate their competition in the area, right? And they enable then themselves to have the resources that those other organisms would be consuming. So again, secondary metabolites, and these don't really necessarily fit neatly into the 4 categories above. You could argue that caffeine and nicotine are similar to the nucleic acids in some ways, but ampicillin doesn't really fit into any of those sorts of things if you look at it. Maybe it's kind of like a peptide. Some things like erythromycin, they might have sugars on them, they might have components that look like polypeptides, so there's a blending mixture of things you can get. So don't just think we're limited to those 4, but they're the main 4 that we look at. So, visiting a little bit more about biology, you're familiar with the central dogma of biology. I often call it molecular biology to emphasize that we're talking about molecules and the way they interact. And you've seen this before. It's a conceptual map that shows how sequence information kind of flows through an organism. And, DNA is, of course, the master library, sacred holder of the genetic information. And that information ends up in RNA, which ends up in protein. And you, of course, recall that the process by which DNA is converted into new DNA is replication, and the process that the information stored in DNA is given to RNA is transcription, and then finally the process of converting that information into a protein is translation. However, I actually like to add another layer to this. I'm going to draw a dotted line because they don't really put this in books. But I think it's important to think about that ultimately we're talking about the organism. And then this arrow here is protein function. This highlights that proteins, it's essentially where the rubber meets the road, if you want to put it that way. It's where those functions are carried out, whether it's a structural protein, whether it's myosin enabling you to flex your muscles, or maybe some kind of ion pump in neural tissue to enable you to think. So it doesn't really stop with proteins, right? But proteins are where that information is stored, and DNA ultimately leads us to protein function and the organism emerging from that. But we are a chemistry course, and I want to be sure we understand bonding to start with. And you might remember the processes of hybridization and this so-called valence bond theory. Just a brief introduction or review of that. So here, let's look at C2H4, which you would identify in IUPAC nomenclature as ethene. Notice there aren't enough hydrogens to fully saturate the carbon, so you can assume that there's going to be a carbon-carbon double bond. So the Lewis structure of ethene would look like this. And then you sort of wonder, we can see this carbon-carbon double bond, and we draw that with 2 lines that look the same. Are those actually the same? And to understand that, let's explore what these types of bonds are. Of course, you remember hybridization, and the carbon, in this case, is going to be sp2 hybridized because it's bonded to 3 other things. And for example, then when we form a bond, we have this sp2 hybridized orbital from one carbon overlapping with the s orbital from hydrogen, creating a bond here. And then we have 2 hybrid orbitals from each carbon overlapping there to create that bond. Now, those are both called sigma bonds. And so when we talk about a double bond, right, one of those bonds is indeed the sigma bond. However, the other bond, it looks the same up here. We just draw 2 lines that look identical, but in fact, the other bond is very different. It's a pi bond. And it's the leftover, the unhybridized p orbital from each carbon, and the overlap you get between those. So then a double bond consists of that sigma bond, but also a pi bond. And a triple bond would have yet another pi bond. But the key thing here is that we get no rotation. Right? It's not going to freely rotate around this double bond, and that's going to have important consequences later on when we talk about things like protein structure. So we put these atoms together, they make bonds, and we can measure the bond length, and we use a unit called angstroms. And if you're not too familiar with what an angstrom is, 1 angstrom, capital A, little circle over it, equals 10 to the minus 10 meters, right? So very, very small. Of course, we're talking about the distance between 2 atoms. Angstroms are convenient because the unit kind of puts these distances 1 point something for the most part. Now, notice I have these all to the hundredths place, and I think if you were to memorize all of these to the hundredths place, you'd be in great shape for an upcoming quiz or exam. Actually, no. If you memorize all of these, you're going to be wasting your time. Please don't do that. I didn't put these on here for memorization. I put them on here, one, because we might use them for an assignment later on. But the other thing is, it's the trends that matter, right? Notice the difference between a carbon-carbon single bond and a carbon-carbon double bond. That it shrinks when you have a double bond because you have more electrons tying those nuclei together. And also when you have things like a carbon bonded to something like an oxygen or a nitrogen, notice that those also shrink because those are more electronegative atoms as well. So it's again, understanding those trends, not memorizing numbers. That's not what's important here. Let's talk about shapes and geometry, and very important in biochemistry since a lot of processes, proteins interacting with small molecules and other interactions happening. If we think about a central atom A bonded to 3 other things, if that's all that's going on, this is going to have a 120 degree bond angle, and it's going to take on the trigonal planar shape. Now, if, for example, this that central A atom is actually a carbon, and maybe it is double bonded to an oxygen, and we have these other groups, whatever they happen to be, right? Notice that this carbon here will have sp2 hybridization, right? Because it's bonded to 3 groups overall. And then another thing we could have, let's add a 4th group bonding, and so we'd have this structure here. We'd have about 109.5 degree bond angles, or maybe 110. And the shape of this molecule, as shown, is tetrahedral. And if we had something like methane, CH4, if all of those X's as shown were in fact hydrogens, then this carbon would have sp3 hybridization. But another possibility could be maybe one of those groups is not the same. It's not like the other. So maybe this is instead an electron lone pair instead of a bonded atom. And an example of that might be something like ammonia, right? NH3 with a lone pair. And that's going to end up changing things a little bit. It's not going to have a true tetrahedral shape anymore because you removed a bonded atom. Instead, it's going to have a trigonal pyramidal shape. And in fact, most of these 3 that we've just looked at, that describes most all of the molecules that we're going to look at in biochemistry, believe it or not. So we're not going to get into seesaw and all these other things. But I will bring up adding a 5th ligand or a 5th group just because we'll see it once or twice later on in this semester. And so now we have a distinct difference. We have 5 things bonded. We actually have distinct axial here and here versus equatorial groups there and there. And so this is 90 degree when we're going between an axial and equatorial group. And then between 2 equatorial groups, it's 120 degrees. And so we call this then overall trigonal bipyramidal. And we're really only going to encounter this when we talk about phosphate chemistry. That's going to be later on in the semester. So if you're not as familiar with that one, that's okay. But please do understand how shape and bond angles and hybridization, all of that stuff is related. So when I said sp3 versus sp2, hopefully you understood that. If not, please do review those. Another thing you might need to review if you haven't thought about these in a little while are the functional groups. These become very, very important when talking about everything we're going to talk about. Protein structure, DNA structure, metabolism. Definitely have to understand those. So, for example, if you remember alkanes, is kind of a repeating CH2 group over and over again, but it could be branched, so that might not be the case. But the key is we got carbon single bonds repeated throughout the structure. That makes it a very non-polar substance because CH bonds and CC bonds are very non-polar. We could also have alkene, and now we've introduced a carbon-carbon double bond, so whether that's ethene or something longer, whatever. And now we'll get into things that are a little bit more polar. For example, if we have a carbon that is bonded to an OH group, right, that's a hydroxyl or an alcohol. If that carbon is instead a carbonyl, like this, and it's bonded to something else, and at least one of those, right, is a hydrogen, that's an aldehyde. And if we, again, have that carbonyl, but instead of just a hydrogen, it's an OH, now that's a carboxylic acid. All of these have been quite polar for the most part. Ketones a little less polar, because now we've got that carbonyl, and although that is a polar group, it's going to be interacting with probably other carbons, and we don't care what those other carbons are doing. Now an amine, in our case, we're probably going to be bonded to carbon for that amine, and depending on the situation, it might be positively charged, it might be neutral. I'm going to say it's positively charged here because that's the way they're going to be most of the time. And then an ester, right, is when we have, again, a central carbonyl group. One side of that's probably going to be a carbon bonded to something. The other side is going to be an oxygen and then probably a carbon. But there are some varieties to esters that we'll see later on. The amide is kind of the cousin of the ester, right? We got their carbonyl group, probably bonded to carbon on one side, but rather than being bonded to oxygen here, we're actually bonded to nitrogen, and then probably a carbon after that. So it's an amide or amide, depending on who taught you organic chemistry. Next up is a sulfhydryl group, which looks a lot like an alcohol, except that it is an SH instead of OH. So we'd call it sulfhydryl or thiol. Either name is appropriate for this. An ether is a little different because it's a central oxygen now bonded to carbon on either side. Notice that's very different than an ester, so please don't get those two confused. Now these last three you may or may not have encountered much before, but phenol, not phenyl like a benzene ring, but phenol, which contains a benzene ring, and it's probably attached to something else. I'll just put the circle in the middle. The key is it has an OH group, and as we'll see, that ends up being ionizable. Imidazolium, now I don't know if you've seen that one, but again, we'll say it's connected to something, and the way I usually draw imidazolium is branches like this to a nitrogen, and I'm going to go ahead and add an H there. And we're going to finish that off with a carbon connecting the two nitrogens. And we've got a double bond here and a double bond here. Pick up another hydrogen. And overall, we have a positively charged structure like this. And this is going to be present in some amino acids that we're going to look at. Another one that we'll see is guanidinium. For guanidinium, we'll probably have carbon, but the key point is we've got a nitrogen, a central carbon, another nitrogen, and another nitrogen. These are going to be NH2 groups. This one's going to be an NH, and then you have a resonating double bond, which creates, again, positive charge. Guanidinium is what this is. It's present in an amino acid and also in a nitrogenous base in DNA and RNA. Some of these groups are ionizable near physiological pH, and we're going to spend a lot of time with that, thinking about what the charges and what the ionization states are. So these would be things like carboxylic acid, amines, sulfhydryls, what else? Phenols, and also imidazolium as well. A lot of times it's called imidazole if it's neutral, but when it's positively charged, we call it imidazolium.